Every time one of your cells decide to divide, it's making an irreversible decision. Cell cycle checkpoints work like binary switches, all or nothing. And once a cell commits to a phase transition, there's no going back. So how does a cell actually make that call? By the end of this video, you'll understand the exact molecular switch your cells use to decide when to divide. And it's one of the most heavily tested mechanisms in MCAT cell biology. We're covering four things today. The four phases of cell cycle, the CDK-cyclin partnership that drives them, and how that partnership gets switched on and how it gets switched back off. Quick orientation first. The cell cycle has four phases. G1, which is the growth after division, and as you can see here, this is basically, if you consider this after the division, this is G1. S phase, which is the DNA replication if growth factors are present, and it's really important to have this growth factor present. G2 is this one right here, a second round of growth before division. And M phase, this one, M phase, is basically when we have the mitosis and cytokinesis where the cell actually splits into two. Now, what actually drives a cell through these phases? Cyclin-dependent kinases or CDKs. Here's the key detail the MCAT loves to test. CDK levels stay constant throughout the entire cell cycle. What changes is their activity, and that's controlled by cyclins. And as you can see here, this is the cyclin here, and this is the CDK. This is the active. I'm gonna go over it in the next graph. So, different cyclins gets made at different times. G1 S-cyclin and S-cyclin rises to trigger entry to S phase. See here, we do have the spike here, and that's the start of G1 with the G1 CDK, and then when we have the spike in the S-cyclin, we having the transition from G1 to S phase. And that's basically how we have it. M-cyclin, though, gets built up through G2 to trigger entry into mitosis. And as you can see here, in the G2, we do have that M-cyclin concentration getting built up throughout the G2, and then when we have that spike, and then when we have the highest concentration of the M-cyclin, we're having that transition from G2 to M phase. This is the G2 and this is the M phase. And it is also important to know, we're going to talk about it later on, but when we have the drop in the M-cyclin though, here, we're going to have that metaphase, anaphase transition. So, H-cyclin binds CDK and targets it to a completely different set of substrate protein. So the cyclin is what gives the CDK its specificity. Same enzyme, different job, depending on which cyclin is attached. Now, we do have a thing called two-step activation. Here's how the activation actually works mechanically. CDKs have a built-in autoinhibition, a T-loop that physically blocks the active site. Here's, for example, is the active site here, and as you can see here, and this is the T-loop. Cyclin binding partially displaces that T-loop, so the CDK becomes partially active, but full activation is the second step. This is the partially active, this is inactive. CDK-activating kinases, or CAK, is the next step that we need. Or the CAKs phosphorylate the CDK to lock it into a fully activated state. And as you can see here, this is the fully activated state of the cyclin, CDK-cyclin that we have. Now, we know how to activate that, but there should be also inhibition part. There should be something that would turn it off. There should be a break. Here's how the break system works. Wee1 kinase can add an inhibitory phosphate, this guy here, that shuts CDK activity down even with cyclin bound. And as you can see here, this is a cyclin. It's bound, but it's inactive. and this is active. CDC25 phosphatase, as you can see here, phosphatase, remove the phosphate, removes that inhibitory phosphate, flipping the CDK back to its active site. This push-pull between the Wee1 and CDC25 is actually how cells create those sharp switch-like or all or nothing transition. And for M-CDK specifically, active M-CDK hyperactivates CDC25. We're going to talk about it later on, but M-CDK hyperactivates the CDC25, suppresses the Wee1 through positive feedback, which rapidly amplifies the signals once the cell commits to entering to the mitosis, and we do have it right here. But anyways, we're going to talk about it later on. Now, the activation is only half of this story. The cell also needs a way to shut this off, or it would just keep dividing. That's basically what causes cancer. But again, we're going to talk about it later on. That's the job of APCC, the Anaphase-Promoting Complex, or cyclosomes. APCC is a E3 ubiquitin ligase. It tags cyclin with a chain of ubiquitin molecules, which marks them for destruction for proteasomes. And this is the chain that we're talking about. We do have the CDC25, we do have the APC, and then we have this guy here. And then if you want to look at the actually how this is the active APC, if you want to call it active APC. See, and then we're going to have an effect here, and then that is going to put M-cyclin for degradation. Now, we talk about the M-CDK. So once M-cyclin gets degraded, M-CDK activity collapses, and that's what allows the cell to go to complete mitosis. Sister chromatids can separate, and cytokinesis can begin. APC is required for the metaphase to anaphase transition. And you can actually see what happens when the system breaks in yeast lacking a functional APCC component. This is the wild type. We have like a normal thing, and then this is the mutant part. Cells fail to complete division properly, and you see abnormal accumulation of multiple cell bodies stuck together instead of clear separation. And then where you can see that here. There's no clear separation. These are the budding yeast. These are just like, yeah, they're not big enough to get separated, so that's why. Now this raises the obvious next question: what actually stops the cycle if something wrong? So like unreplicated DNA or chromosome that are attached to the spindle. That's the checkpoint system itself, and that's coming in the next future video. And all right, quick question: if cell's APCC is non-functional, what happens to the M-cyclin levels, and what's the consequences for the cell? Want to guess, or commented below? M-cyclin cannot be degraded, so M-CDK activity stays high. It's not going to go down, it stays high. And what happens? The cell cannot complete the metaphase to anaphase transition, and it gets stuck, unable to finish the mitosis. And as you can see here, we have that metaphase to anaphase when we have that drop in the M-cyclin levels, and that's why I told you to remember this part, and I said we're going to talk about it later on in the video. So here's a recap. CDK levels stay constant. This is important. CDK levels stay constant, but cyclin controls their activity and specificity. Cyclin binding partially activates CDK and CDK. CAK phosphorylation fully activates it. Wee1 and CDC25 fine-tune that switch, and APCC shuts down the whole thing by tagging cyclins for destruction, resetting the system for the next cycle. If you liked the video, please like and subscribe. Tomorrow we're back on Organic Chem. See you then.